Hi, I'm Amanda.
I'm Abby. Amanda and I are studying genes that cause repression in grasses, specifically in the leaves and flowers.
We hope that by better understanding the function of these genes, we can use it to increase crop yields, which will help feed the rapidly growing human population.
Hi, I'm Kirsten. I'm a high school intern looking to explore the molecular underpinnings of plant diversity through studying awns in Brachypodium and B-class genes in maize.
Hi, I'm Benedict. I was an MLSC intern in the lab last year, and this year I was rehired through research money as an undergraduate, since I'll be a biology major at UMass starting this fall.
We work in the Bartlett lab studying plant evolution and development.
We were allowed this opportunity by the Massachusetts Life Sciences Center in the Collaborative, and we'd like to thank them for allowing the lab to pursue our goals.
Just a note, all of the plants shown have naturally occurring mutations and are not genetically modified or transgenic.
Right, so we're out in the field. Here is a drooping leaf double mutant. So since two of its genes are knocked out, it just doesn't produce a midrib in its leaf, and so its leaves are drooping. You can see the phenotype there. Whereas in the wild type, it has a very pronounced midrib that will break if you bend it, and you can see how the phenotype's affected by how we mutate the plants.
This is a very normal corn tassel. Gotta take a look at it. So this is a wild type, but like this is the tassel seed mutant. As you can see, there are corn silks growing out of the tassel, and it started growing corn kernels up top too. Here we have a grassy tiller mutant, or a GT mutant. If you compare it with the wild type, which has one singular stalk growing up from the ground, the GT mutant has many little stalks that are called tillers. One of our many field tasks to complete was shoot bagging. It ensures that the ear is pollinated correctly when we make specific crosses between corn plants.
The first step of shoot bagging is, of course, to find the ear that you want to put the bag on. We usually just pollinate the top one, so if there are multiple, you can just ignore the ones that are lower. They're usually hiding behind the leaves here. Now this one looks about the right size. You want to make sure that it's big enough so that the bag will not fall off of it. You take the side that's longer, the side that does not have text on it, and you put that against the stalk. And you want to put it over the ear, making sure that you're not missing any of the leaves. So you put it down, nice and tight. You check the front and the back to make sure that you haven't missed any leaves. And then move on to the next one.
Right now, Kirsten is setting up a polymerase chain reaction, or PCR. The Bartlett lab does a lot of PCR. It is a cheap and relatively simple method for amplifying the DNA sample to be studied. This is incredibly useful in many ways. For example, the DNA can be run out on a gel, which can reveal plant genotypes. The DNA can also be used in other experiments. And here, Abby and Adriana are doing a bacterial transformation. Previously, they took a plasmid, or piece of circular DNA, and heat shocked some E. coli cells so they would take up that plasmid. Right now, they are pipetting the E. coli onto plates and using shaker beads to spread the bacteria around. Later, they will put the plates in an incubator, and tomorrow they will collect colonies and grow cultures in order to harvest a large quantity of the plasmid.
We've had a great introduction to advanced lab work, and this internship provided us with an invaluable opportunity to contribute to science.
We've learned many protocols, lab etiquette, time management, and a lot of problem solving.
Our time in the lab will influence our lives and the lives of those who benefit from our work for years to come.
We are incredibly grateful to Dr. Bartlett for this amazing opportunity, and to her postdoctoral and graduate researchers for mentoring us.
Thank you!